Historical landmark in virology and immunology. In this chapter, we will briefly point out the key important historical landmark in the field of virology and immunology to know more about the events in history of microbiology. Both virology and immunology is a branch of medical microbiology. The virology deals with the study about viruses and immunology study about the immune system that protect our body from harmful microorganisms and foreign particles that cause infection or disease. Discovery in virology. By the late 19th century, many diseases were linked to bacteria, but some like smallpox, measles, and the common cold has no known bacterial cause. Pastors suspected that the rabies agent was too small or just smaller than bacteria to see under microscope, and that is responsible for the infection like smallpox, measles, and common cold. In 1892, Ivanovsky proved the extensive of ultra-microscopic microorganism by transmitting mosaic disease in tobacco plant using bacteria-free juice from diseased leaf. Bergering confirmed that this in 1898. In 1898, Loeffler and Fronch showed that the filterable agent caused foot and mouth disease. Yellow fever was first human disease proved to have a viral cause. Walter and Reed confirmed its transmission through infected mosquito bite in Cuba at 1902. In 1934, Ernest Ruka developed the electron microscope. With the help of this electron microscope, detailed examination of smallest virus can be seen through this electron microscope. Because viruses are way smaller than that of bacteria, it is measured in nanometer. But in the case of bacteria, it is measured in micron or micrometer. So in the simple or compound microscope, virus cannot be seen. Only through the help of this electron microscope, we can observe the structure of the virus, particle or virus. In 1913, Good Pasteur developed the chick embryo technique for virus cultivation. And this chick embryo is used for the cultivation of virus. And if you want to cultivate the virus, we will inoculate the virus into chick embryo and we will keep it for incubation. After that, we can isolate the virus from the chick embryo. Ellerman and Bank in 1908 were the first to suggest that viral infection could lead to malignancy. Malignancy means cancer. That means viral infection will lead to cancer. In 1911, Rose isolated a virus causing sarcoma in fowls. Then sarcoma in fowl means the cancer in fowl. Towart in 1915 and D. Hurley in 1970 independently discovered bacteriophages. Bacteriophages are virus infecting bacteria are called as bacteriophage. In 1796, Edward Jenner successfully immunized against smallpox using the cowpox virus. He observes that milkmaid exposed to cowpox were immune to smallpox. That leads to the development of vaccination technique. In 1888, George Nuttall and observed that defibrinated blood has a bactericidal effect. Bactericidal effect means killing of bacteria is called as bactericidal. So those agents which will kill the bacteria, we will call it as a bactericidal agents. In 1889, Edward Butcher discovered that heating sera for 1 hour at 55 degrees Celsius destroyed its bactericidal effect. Next, we go for the important historical landmark in immunology. In 1883, Paul Ehrlich and McIntosh proposed that the phagocytic response is the primary defense mechanism against microorganisms invading blood and tissue, establishing the concept of cellular immunity. That means phagocytosis response means it is an immune response produced by our own body against the microscopic or antigen or external organisms. In 1819, von Behring and Kittastor identified a special humoral factor or an antibody produced in guinea pig serum by injecting serial sublethal dogs in tetanus toxin. In 1893, Richard Prefer demonstrated the bactericidal effect in vivo by injecting live cholera vibrio into specifically immunized guinea pig showing bacteriolysis. And in 1895, Julius Bodet established that bacteriolysis in vivo required two factors, a heat-stable antibody and heat-labile factor, which Hannes Bucher in 1889 called alexin, and Ehrlich later named this as complement. In 1903, Wright described opsonin in serum. Opsonin is a substance which will go and bind to the microorganism or antigen, which will enhance the phagocytosis process. The discipline of allergy and its role in disease pathogenesis emerged from Porter and Charles Richard. In 1902, experimented on dogs, which leads to the discovery of anaphylactic reaction. In 1914s, Medver et al. Established the immunological basis of transplantation. Their work, along with Bernard's, enabled successful transplant through elective immunosuppression and donor selection based on histocompatibility. In 1967, Bernard proposed the concept of immunological surveillance, suggesting that the immune system primary function is to protect our body by destroying foreign antigens, including all type of microorganism, including malignant cell also arising from somatic mutation. And actually, this malignancy was viewed as a failure of this immunological surveillance. In 1995, Niels Kajor proposed the natural selection theory of antibody synthesis, which Bernard modified into clonal selection theory in 1957.
Subscribe to Simply Micromed. If you're curious about medical microbiology, infectious diseases, and unraveling the mysteries of tiny organisms, hit that subscribe button. Join our community of science enthusiasts, and let's explore the microscopic world together. Remember, knowledge is contagious. Spread it by liking and subscribing. Thank you.